So, good evening. Today is our 12th session. So, this is our last session. So, in this, we'll be trying to address more and more questions which you have regarding this course. And along with that, we'll cover the content related to week 12, so which was related to pathway enrichment, gene set enrichment analysis, LinkedOmics, and proteogenomic opportunities and challenges. So, in this, we'll be practicing MCQs and a little bit about the current literature and literature related to these softwares and tools. And along with that, I'll take your doubts and we'll be discussing previous year assignment questions of week 12 topics. So let's start with our first question. So the first question is, what new programs were created after the Clinical Proteomic Tumor Analysis Consortium, which is also known as CPTAC, Progress in Proteogenomics? So option A is Applied Proteogenomics Organizational Learning and Outcomes, Apollo. Option B, International Cancer Proteogenome Consortium, ICPC, and option C, TCGA, which is the Cancer Genome Atlas. So from these four options, you have to select that which of these programs were created after the progress of CPTAG. So any guesses? Okay, so now let's see what would be the right answer. So the right answer is option 3, which is A and B. So Apollo, which is a program created to integrate the proteogenomics into the cancer treatment for real-time clinical decisions. So this was created after CPTAG, and then International Cancer Proteogenome Consortium, which is an international collaboration formed to expand the proteogenomic research globally. So this was also created after the establishment of CPTAG. But when we talk about TCGA, so it's related to genomics, not proteogenomics. So it was established earlier than CPTAG. So this is the one which is not created after CPTAG, but it is created prior to CPTAG. So hence option 3, which is A and B, was the right option. Now let's move to the next question. So the next question is, what new efforts are being done by CPTAG in the field of cancer proteogenomics? Option A, characterization of additional cancer types. Option B, expanding its application through open source community resources. Option C, accelerating precision oncology by applying proteogenomics to questions of toxicity and resistance in clinical trials. So in this you have to tell that what are the efforts which are done by CPTAG. Yeah. So the right answer is option 4 because these all are the efforts which are done by CPTAG. So all these options are right. Now we'll talk about what is gene set enrichment analysis and how this approach was developed. So this was the first publication which came up in 2005 related to the GSEA. So it is a knowledge-based approach for interpreting genome-wide expression profiles. So as it is clear from its name that it is a gene set enrichment analysis. So it means that you are kind of creating a set of genes and you're studying their enrichment. So that enrichment could be either could be functional enrichment or positional enrichment. So it depends that what enrichment you are studying. So that analysis and why we are performing it to interpret that after getting the genome-wide expression data, we try to interpret that what can we understand or infer from that data. So for that reasons, this GSEA was established. So it is a powerful method for interpreting the gene expression data. So keep this thing in mind that it is for the gene level analysis and it takes gene expression data. So in that way, it addressed the challenges in extracting biological insights from genome-wide RNA analysis. So it will be a genome-wide analysis and the data set can be obtained of any expression level. So in this case, it is the RNA analysis and it will give you the results at the level of which genes and not exactly the transcripts or proteins. So keep that thing in mind and it focuses on the gene sets, which are group of genes sharing common biological functions, chromosomal locations or regulatory mechanisms. So these are different ways to group your genes, so which could either be a biological function, which could be its phenotype, or which could also be its chromosomal locations or some regulatory mechanisms. So these are the characteristics on the basis of which you can means cluster your genes or you can divide your gene into certain sets and you can study their enrichment. So, but the major or the most by default used criteria is to look for the biological functional level significance, that what is the functional level similarities between them, and then characterize them into different gene sets. Then GSEA provides valuable insights into cancer-related datasets, including leukemia and lung cancer. So in most cases, when GSEA is used, so it is mostly used for the cancer dataset analysis, because in that way it is easier to understand that data and interpret that data so that we can design some therapeutics. So in that way, it is mostly used for the cancer-related datasets. And then the method reveals shared biological pathways, even when single gene analysis show little similarity between studies, such as patient survival in lung cancer. So, in this way, if the criteria of dividing it into gene set is the biological pathways, so in that way, that analysis can also be done with the help of GSEA. And it is also available as a free software. So that means anybody can access it and use it to interpret their data, and it is accompanied by a database of 1,325 biological defined gene sets. So these gene sets are already defined in GSEA, which you will be able to find. Now we'll look into the key elements of GSEA, that what are the key elements. So such as enrichment score, which is the most important element of GSEA because it assigns enrichment score to the gene sets. So enrichment score is calculated to see that how much a gene set is overrepresented at the extremes of a ranked gene list. So what it does is it ranks the gene list which is provided by you. So means in the gene list you will be providing the gene IDs along with their expression profiles. So what is the expression of this particular gene in the sample? So in that way it will try to rank that genes. So in that way, first step is to rank that gene list, and then that score is tracked by moving through the list, increasing it for the genes in S and decreasing it for those not in S. So in this case, S means gene set. So while it will categorize those genes into different sets, then it will move those genes up and down in the list depending on their presence either in the set or they are not present. So in that way, it will decrease the scores. If that is not present in the gene set, then it will decrease the score for that gene. And if it is present in the gene set, it will increase that score. So the highest score we reach during this process indicates that how enriched the gene set is. So in that way, it will provide the enrichment score for the gene set. Now estimate significance of ES. So now after calculating the enrichment score, next thing is to estimate that whether that enrichment score is significant or not. So in that case, they determine that how the statistically significant the enrichment score is by permuting the phenotype labels and recalculating the ES for these shuffled data sets. So now whenever enrichment score is calculated in the first run, so it's not always that it will always be significant. So in that way, again and again, scores have to be verified and it has to be checked. So in that case, for establishing that significance, it performs the permutations. So after permutations and looking for the phenotypic labels that either those labels are associated properly or not, so it does the recalculation again and again. For that, it will try to shuffle the data sets. So it will shuffle the data set and each time it will change the enrichment score until and unless it find it to be statistically significant. And those significance values are either defined by the user or there is already predefined statistical significant value, which means that if p value is less than 0.05, then that means the results are significant. So in that way, it performs again and again shuffling of the data sets until that significance value is reached. So this creates a null distribution to compare the observed score against, giving a better assessment of significance while preserving the relationship between genes. So in that way, it will compare those distributions. So in each time ES will be checked, so it will create a null distribution and it will compare the new ES score with the previous ones, and it will check that whether the genes which are present in each set, whether they are correlated or not, whether they are getting disturbed or either it is preserved. So that thing is done in the case of significance of ES and then adjust for multiple testing. So now after checking for the significance, next step in any statistical method is to adjust for multiple testing. So which we have seen in the previous sessions also, that there are some other tests which comes after the statistical tests, so which is known as multiple testing corrections and all other things. So when analyzing many gene sets, so in case we have multiple gene sets and we are trying to establish the significance, so in that case we have to adjust for the multiple testings so that it could be significant. So in that case, it normalizes the enrichment score for each gene set based on its size. Because, for example, if a one gene set is composed of multiple genes, for example, 100 gene, and other gene set is composed of 7 genes. So in that way, it doesn't make any sense that you compare their scores, their enrichment scores, without normalizing them, because there is a bias in the number of values which are present in each set. So in that case, we need to normalize it so that those biases can be corrected. So in that case, when there are multiple gene sets, we need to account for that multiple comparisons. So in that case, we need to normalize the enrichment score for each gene set based on its size. And then we have to calculate the false discovery rate to estimate the likelihood that a significant finding is false positive. So all those criteria are checked, that whether those positives which are identified, either those are false positives or they are true positives. So all those adjustments are done, and in the end, enrichment score is produced. So is it clear? Because these are quite complicated terms. So for ease, I would say that you might have seen during using many tools, they give an option of how many times you want to run the batch, number of batches, or you can say it asks you to for the number of runs, that for how many runs you want to give your analysis for. So in that case, those runs are nothing but they are for adjusting for the multiple testing only. They are just trying to rerun your, I mean, they are just randomizing your sample set and again running it so that in the end, the results which are produced could be more significant. So in that case, this is the main crux of GSEA, that how it gives enrichment score and how it makes sure that that enrichment score is not generated by chance. It is a significant score, and ultimately, if there are multiple gene sets, then we also have to go for the multiple testing corrections. So this is all about GSEA. And if you look at the main homepage of GSEA, then it appears like this. And first of all, it is developed by the Broad Institute in UC San Diego, and this is a computational method which determines whether an a priori defined set of genes shows statistically significant concordant differences between two biological states. So in this, there will be two different samples. One will be from the normal state and another will be from the cancer state or disease state. It depends on you that if you are studying the phenotypes, that either disease and normal phenotype, then it will be sample A and sample B, and then you will be comparing that what are the differences among them. So we have already seen there are multiple statistical tests which are often chosen to determine that either two sets are means varying enough or not. So all those tests are available already. So this GSE also does that. It already have predefined statistical tests, or either you can also select them. So in that way, just try to give you that which genes are enriched in your sample. So that you can compare them, and in that way you'll be able to know that which genes are overexpressed or which genes are underexpressed during cancer state. So those comparisons will be made, and ultimately you'll be able to understand that which genes are important in the cancer phenotype, so that you can target those genes and design therapeutics which specifically target those genes. So that is the main purpose of GSEA, and this is all about it. So you don't have to worry about this background about enrichment, how those calculations are done, but this was just for your understanding that what is done mathematically in this server. So this is all about GSEA. And now I'll be having few questions related to GSEA only. So question number 3 is that what does GSEA use to rank genes in a list? So in this, you have to select one which appears to be more appropriate. So option A is their chromosomal location. Option B, their correlation with the phenotype. Option C, their protein-protein interaction strength. And option D is their sequence length. So it is actually a MCQ, which was there in your previous year assignment. But yeah, it should be a MSQ, but okay. Yeah, so if it is a MCQ, so right option will be option B, which is their correlation with the phenotype. But other than this, their chromosomal locations as well as their protein-protein interaction strengths can also be used to some extent. But if we have only the expression data, so in that case, we won't be able to get the protein-protein interaction strength. Whereas we can classify them on the basis of their chromosomal locations. But in case if we have two different samples, then in that case, if we are comparing those genes, so in that way, the best technique which is used to rank those genes will be their correlation with the phenotype. So in this, option B is the right. And now question number 4. So in GSEA, what does a high enrichment score indicates? So as we have discussed about in enrichment scores, so enrichment score can either be high or it can be low. So what does it indicate? Option A gene set is evenly distributed in the rank list. Option B, a gene set is concentrated at the top or the bottom of the ranked list. Option C, the dataset contains too many genes. And option D, no association between the gene set and the phenotype. Yeah, so option B is right because enrichment score, if it is high, which means that there are certain genes which are either concentrated at the top or at the bottom of the rank list. So that's why we are calculating enrichment score because we want to see that what is the difference among different genes. So option B is right. Question number 5. So what method is used in GSEA for multiple testing correction? Because we have seen the third step is the multiple testing correction. So option A is Benjamini-Hochberg procedure, Bonferroni correction, permutation test, and Z-test. So these are few of the options for the multiple testing correction, and you have to select one method which GSEA uses. Okay, so we are confused among both of them. So the right answer is option A, which is the Benjamini-Hochberg procedure. So actually, these all are multiple testing correction methods except few of them. So, but we'll see that why Benjamini-Hochberg method is generally used. So it controls the false discovery rate. It balances the true signal detection with minimizing false positives in GSEA. So Benjamini is most of the times it is selected over other methods. But when we talk about Bonferroni correction, so it is too conservative for large data sets. So if your data set is large, then you cannot go for Bonferroni correction because it might reduce the ability to detect the true positives. So in that case, you do not opt for Bonferroni. But if your data set size is small, then you can easily go for Bonferroni correction. Otherwise, if data set is large, then we select Benjamini-Hochberg procedure. If we talk about permutation test, so it evaluates the significance of enrichment score and it's not for the multiple testing correction. It is at the second step of GSD, which is for the significance of enrichment score. And if we talk about Z-test, then it compares the mean between two groups. So this is also at the second step only when we are comparing between two samples that either they are different from significantly different from one another or not. So in that way, it is also not for the multiple testing correction. So the right option is option A. Now we'll talk about LinkedOmics. So this was also another tool which is related to study gene set enrichment analysis, and it is combining actually multiple omics data. So it is for those people who have multiple data sets and they want to analyze it and they do not have that much expertise in the computational analysis. So this is a combined platform which provides you to look into huge properties of those data sets and combine multiple omic analysis. So in this case, this paper was published in 2018 in Nucleic Acids Research, and it tells that linked omics can be used to analyze multi-omic data within and across 32 cancer types. So in that case, when it was established, they already have studied it on 32 cancer types, and about 11,000 patients data was taken as a part of TCGA project. So from TCGA, they have collected that clinical and multi-omic data. So it is the first multi-omic database which integrated mass spectrometry-based global proteomics data from CT-tag or on the selected TCGA tumor samples. So in that way, they have combined two different data sets. One is TCGA, which is at the level of genome, and then which is a mass spec-based and proteomic level data from CP-tag. So they have combined both genomics and proteomics data sets. So this is kind of proteogenomic level analysis and it contains more than a billion data points, which allow for the comprehensive analysis of multi-omic cancer data. So that means there are multiple things multiple properties or multiple fields you can study about the genes or the data set which you provide. So there are three main modules which are used for analysis. So first module is link finder. So link finder, what help it provides you? It enables the flexible exploration of associations between molecular or clinical attributes and also analyzing and visualizing billion of attribute pairs with cancer cohorts. So in that way, it is for the association studies. When you are trying to look for associations, when you have a sample data set, so you are looking for the how means what are the association between different gene set or molecules or clinical attributes. So in that, along with that, you also analyze and visualize them. So you can also visualize with the cancer cohorts. So in the later slides, we'll see that how linked omics helps you to visualize that data and how associations are generally observed and what other attributes that you can study. So when we say attributes, they are nothing but they are just features, either survival or expression. So all those things come under attribute pairs. Then comes link compare. So this is for the comparison of association across different cancer types. For example, if you are studying for the breast cancer as well as for the another type of cancer, ovarian cancer, so in that way you can also compare those data sets together. So compare is for if you have more than one data set, then you can compare among them. And then comes link interpreter. So it transforms the identified associations into biological insights. So after you have performed that analysis, so it will also associate other terms to it, such as pathway information or network analysis information. So in that way, you can better interpret your results. Then demonstrated through 5 case studies that linked omics is a valuable platform for biologists and clinicians to access and analyze cancer data across different tumor types. So they have performed 5 case studies, and it is also freely available at this particular link. You can easily access LinkedOmics. So this is their workflow, that how it works. So first of all, from TCGA, they have collected the data of different cancer types. And from CPTAC, they have collected the information at the level of proteomics, which is mass spec based data. And then they have used the link finder, which will ultimately look for the associations within and cross omics associations. Then link interpreter will also associate the information such as pathway network analysis if you have only a single data set, such as single cancer type. But if you have multiple cancer types, then you can go for link compare and you can compare them. And along with this, you can apply link interpreter on both those data sets so that you can also look in what pathways those genes are involved and what are the phenotypes and perform network analysis. So in this portion, they have given the analysis which they have performed. So as they have told that they have 32 cancer types and these many patients. So this is a visual representation that these are the cancer cohorts. So cancer cohort means there are different cancer types. So in that way, those small group of people, which we have also previously seen, that cohort means that small group of people belonging to certain type which are studied for the clinical implications and different studies which are performed on them. So it is just a group. And in that way, number of samples and cancer cohort, they have plotted it that which cancer is more frequently observed. So means this will tell you that in how many means, for example, there are certain cancers which are majorly observed as compared to other cancers which are not that much observed. So this is just that representation. And then comes 100,000 molecular attributes, and that they have told about different attributes. So in this case, attributes could either be clinical attributes or methylation, miRNA, mutations, so single cell nuclear antigen receptors. So all those things, these are kind of properties which they have studied. So these are attributes and these are their cancer cohorts. So they are telling that in which cancer cohort, what are different attributes that can be studied. This is also just for the representation that how they are able to capture multiple things from different cohorts. So for that purposes, they have kept it. Now they are telling that they can also means we can also visualize our results in the form of volcano plot in which we can see the signal strength and we can also compare the p-values that either those means signal strengths are significant enough or not. We can also visualize the results in the form of scatter plot. So in that case, they have taken one example of this particular gene ERBB2 and they are looking for the copy number versus phosphosites. So means how those two properties are linked with one another. So in that way, this is one of the plot. Then we can also go for the box plot representation. So mutation status and what is the RNA expression. So means either mutation is influencing the expression of that particular gene or not. So that comparison can also be done. Then come survival curves. So in this, we can also even study that how survival is influenced. For example, if there are mutations in one particular gene, then either it will lead to the better prognosis or it will means affect the individual and it will decrease their survival abilities and it will decrease their lifetime. So in that way, survival curves are also important. Then gene ontology enrichment. So this comes under link interpreter in which we can also associate gene ontologies or pathway enrichment information to our data set so that we can study that in which pathways our genes are involved or which pathways are majorly altered. So as we have seen in multiple literature that during the cancer phenotype, which are the pathways which are mostly altered are the either cell cycle involved genes, or they are either related to apoptosis or growth. So these are the major pathways or signaling pathways which are majorly altered. So that means those genes are majorly enriched in the cancer phenotype. So all those analyses are done with the help of link interpreter and then pathway enrichment as well. In link compare, you can, when you are comparing two different cancer cohorts or cancer types or samples, so in that way you can go for scatter plot, or you can also go for Venn plot. In that way, you can compare that. For example, there are certain genes which might get mutated in both the BRCA, means breast cancer, as well as ovarian cancer. So that analysis you can do, that comparisons you can do, that is referred to as Venn plot, where you are looking for the overlaps, that in two sets which are the genes or what is the expression which is overlapping among two sets. So that is known as Venn plot. And then comes heat map, which you have, I think, seen a lot in the assignment as well. You have to compare the expression among both of them so that you can calculate that how the expression is varying. So among two phenotypes, so among two states, what is the expression of that particular gene? Either it is overexpressed or it is underexpressed among different patients or among different individuals from their samples are collected. So heat map is the representation, visual representation when you are comparing it. So in this case, in the heat maps, instead of writing numbers, we provide the color codes. So in that way, we can visually see that where the expression is varying. And now this is a difference among different modules. So we have already studied them, but the new things which I want to add is about the statistical analysis. So in link finder, we use different statistical analysis or different tests such as Pearson's correlation, because we know that in link finder, we are looking for the associations between molecular and clinical attributes. So to study those associations or correlations, we can use Pearson's correlation test, or we can use ANOVA. We can also go for t-test or Fisher's exact test. So these all tests come under link finder. Along with this, multiple test correction by FDR. So as we have already seen this, so Benjamini-Hochberg test, and then visualization tools. So in this case, results can be either visualized in the form of table, which is known as tabular results, or we can see the volcano plots. Along with this, we can also visualize heat maps, scatter plot, box plot, and survival curves in link finder. If we talk about link compare, so as we know, it compare among two different data types or different tumor types or subtypes. So in this way, meta-analysis is meta-analysis is done using the sums method for consistent associations. So there are some methods. So sums is one among them, which is generally used for the comparison. And then in this case, visualizations will be in the form of scatter plots or Venn diagrams, heat maps, or bar plots. And if we talk about link interpreter, where we are associating insights such as biological pathways or other gene ontology information. So in that case, and one more thing is that it performed that enrichment using web gestalt. So from there also it is taking information of the pathways and other things. And in this case, functional enrichment against 26,449 categories of gene ontology, KEGG, reactome, protein-protein interaction networks. So from all of them, the information is collected for this association, and then it visualizes the enriched pathways and functional categories from multiple biological databases. So in this case, the representation will be in the form of either pathways or in the network form. So this is all about the three modules. Now we'll practice one MCQ. So means practice the MCQs which are dedicated to LinkedOmics. So in this case, we have to follow stepwise. So I don't know whether this question could be asked in the exam or not, because I don't think they'll be providing you the internet facilities. So now let's see, this was in your previous year's assignment. So the question is that RA is working on glioma project, which is a type of brain cancer, and she wants to check the IDH1 isocitrate dehydrogenase 1 mutation status in LinkedOmics. This software can analyze multi-omics data of various cancer types. So you have to follow the given instructions and help Arya to solve her queries regarding the IDH mutation. So these are the steps. So now from this question only, you can get the idea that what kind of analysis you can do. So how the LinkedOmics can help you to answer your queries. So if you are performing some cancer research, so what are the different questions that you can ask? So one such question is that you have the name of the gene and its mutation status, and you are just trying to check that means what are the mutation, means how that mutation is linked to this particular cancer. So similarly, this area is also doing the same. So in this, there are different steps which are mentioned, and we are one by one, we are just move through these steps. So in this case, it won't be like a MCQ thing. We are just following the steps and we'll see. So the first step was that we have to go to the LinkedOmic website. Then we have to enter as a guest, and then in each slide the steps will appear. So we'll see. Now, first thing is, if you go to the this home page of LinkedOmics, so it appears like this. So first thing is that either you can sign in if you have your ID, otherwise you can simply enter as a guest. So in that case, you do not have to sign in. So what are the advantages of signing in? So in case if you are a research scholar or you are a person who are means again and again doing these analysis, then instead of entering as a guest, you can simply sign up and then get your login ID and credentials, because in that way you will be able to save the projects. And if you enter as a guest, then another time, if you want to access that same data analysis which you have performed, then you won't be able to, because it will get deleted. So that's why that is one of the reason that why we can sign up here. So if we have to work again and again with the same data set and perform analysis again and again, then we can sign up. Otherwise, we can enter as a guest. So this was our first step that we have to follow. Now, next thing is that we have to click on this new analysis, which is in the left hand side. So whenever you will click on new analysis, so these things will be appeared. Now in LinkedOmics, you have 5 steps which you have to follow to perform any analysis. So beginning with the first step, which is select cancer cohort. So now you have to select a cancer cohort. So there are multiple types of cancer, as they have mentioned. There are 32 types of cancers which they have taken in this data set. So now we have to select one cancer cohort. So as per our question, the cancer cohort which was asked to us to select was, actually it's not mentioned in the slide. So if you click on the new analysis, then this particular home page, means this particular page will appear. Now what we have to do is, now next step is that we have to search for glioma, which is a type of brain cancer only. So you have to write here on the search box glioma, in case it is, if it is not visible. Otherwise, what another thing you can do is that from here they are asking you show entries. So in this case, you can also select show all entries, or otherwise you can select 5, 10, 50. So that is other options, or otherwise you can simply write it on the search box. That would be much easier option to directly see that. So in this case, we have to select glioma. Now we'll click here. So in this way, it will be selected. Now keep one thing in mind that so in the current year assignment, what they have done is they have not updated it. So what happened is that this particular uh, software tool has updated. So there are many things which are not present currently in this tool. So don't worry about that. Keep those things in mind that many of those previously available things, they have deleted them, which are no longer visible. So now first thing we have to do is we have to select the cancer type. Now after selecting cancer type, it will itself create the step 2. So each time when you means complete one step, it will show you the next step. So you cannot see the all the steps at one time. So means it is step-wise thing. So after selecting one of the cancer cohort, it will give you the next step, which is to select the search data set. So from where you want to search, because we already know that there are different data sets, there are different methods through which you can collect the data. So in that way, we have to select that which should be our search data set. Either it should be RNA and RNA-seq data, or it should be mutation level data, what kind of data we want. So that is our second step. So in this case, they have mentioned that we have to search the dataset as the sample cohort TCGA GBM LGG. In this platform should be GA2X and analysis level should be at the level of G. So now what we have to do is we simply have to show all entries so that it will be easier for us. Now from all of them, we have to see that where this option is matched. So now these boxes also, if you know what platform you have to choose, GA2X, so simply you can uh, click on this box, it will pop up, or simply you can type here GA2X. So in that way, it will filter out other options. So that is also one easier way to see. Otherwise, you can simply, because there are only these few options from all entries, so you can simply find your option here. So in this case, we have to look for analysis level, which should be G. and this should be GA2X, and the platform should be TCGA, GVM, LGG should be the sample cohort. So in this case, TCGA, with this we already have selected here. So it will not be visible here. That is our sample cohort. Now we simply have to look GA2X and GeneSet. So in all these platforms, there are only two options for GA2X, which are this and this. And for gene, there is only this one option. So we'll select this option, which is for the mutation data type. And platform is GA2X. And then analysis level is gene, and description is Firehorse MuteSig 2CV. So during previous times in their dataset, they also used to give the name of the institute, which is now deactivated. So now there is no longer the institute name available. Now third step. So in 2B, we have to skip this step because this is to select the sample dataset. So this is optional. So if you want to select another dataset, then also you can select along with the mutation dataset. Now the third step is to select the search database attribute. So now you have to select the attribute. So in this case, if you have selected the mutation database, so mutation data type, so in this, in case of search dataset attributes, there will be the name of genes. But if you select any other options, then this search dataset might vary. The attributes might vary. So in this case, we have to select IDH1 because they have to study about this particular gene. So in that way, IDH1 will be typed here. Now we will be taken to step number 4. So in this case, we were asked, let's select the target dataset as data type as RNA-seq, platform as high-seq RNA, and analysis level of G. So now one is the sample dataset and another is the target dataset. So that what would be your target? So in this case, this was our option. That means data type should be RNA-seq. And in case you are not able to find it, you can simply write on these boxes. So for data type, you can type here. So in that way, it will try to filter out your search. That will be easier for means finding out if you have a large dataset list. In this case, the dataset list is short, so we can easily get it. So the data type should be RNA-seq and platform should be high-seq RNA. So there are different platforms from where data is collected. So in this case, they specifically want high-seq data, and then analysis level should be at the level of gene because we are looking for the gene level mutations. So that's why this was gene, and this is Firehose RSEM log 2. Description is means this is the description. Now we'll be taken to step 5. So now in step 5, we have to select the statistical method. So in this question, they have asked us to select t-test. Otherwise, there is another test also, which is Wilcoxon-Rank-Sum test, which is also available other than t-test. So from this, we have selected t-test, and now we have to submit our query, which will be our last step of this particular tool. So now after clicking on submit query, what it will do is it will take some time to complete the analysis, and after that, the status will appear as completed. So I think it will take hardly 2 to 3 minutes for this analysis to be completed. Now in this question only, they have asked us to keep all parameters same and wait until the status reach to complete. And now after completion, there will be means different entries which might appear. So in this case, there are only one entry which appears. So now after completion, we need to click on the view option. So this is the view option. Now in this case, there is only one data entry. So we'll click on the view option. We'll click here. Now what you can find in the result section, first of all, these are the association results. So association result, that means it is at the level of LinkedOmic link finder. So there are search attributes. So in this case, firstly, they'll mention the search dataset and the target dataset. So both are same, sample cohort remains same, but the data type of search dataset and target dataset is different. So in search, we selected mutation, and in target, we selected RNA-seq. Analysis level is at the level of gene, and then this mentions the sample size. So in the search dataset, sample size was 799, and in the target dataset, sample size is 669 patients. But there are these many attributes which are available, and they have also looked into the overlap of samples, that how many samples are overlapping among them. So means which are common in both of them. So there are 657 samples which are common, and then there are some other parameters which they have calculated. So statistical test, t-test. So this is a kind of a summary of the results. and this compare is for the link compare. So in case we have more than one entries, if we have two different cohort studies, so in that case we can compare it. So in that way, if we have two entries, then only we can go for compare. Otherwise, with one single entry, we cannot use this compare option. Now we have to click on the view option. Now what it will tell us, so in link finder, so if we have only a single data set, then there are two modules which we can study. One is link finder, another is link interpreter. So in link finder, after clicking on the view option, it will firstly mention you the target genes or attributes. So this is the list of genes, and these are their log2 fold change values. So log2 fold change. So fold change tells you that how much the means expression is variable. So there are multiple means equations through which you can calculate the fold change. So it depends on person to person that how they have developed the fold change criteria. So, but the means by default technique is that you compare among both of them the expression, that what is the expression in the cancer divided by what is expression in the normal. So in that way, if your log fold change is 2, that means your expression is twofold. That means in one state, the expression, the gene is doubly expressed. Or if it is zero point 0.5, that means the gene is expressed in the lower quantities. So it is, and that expression is half. But in this case, if there is log 2, that means you are further transforming your data. So in log fold 2 will be the values which you will get, they will be like a power of 2. So means the value which you are given, when you will put them on the power of 2, then you will get the main expression values. So in that way, this is one of the method to just fit your data into the smaller sections, or in, you can say, to means normalize your data, to transform your data, we have to perform the log transformations and normalizations. So log twofold change, these are the values which tells you that how much expression is variable. Then comes p-value, because this is for the significance. Then comes FDR, which is for the multiple testing correction. We look for the false discovery rate and using the Benjamin-Hochberg method. So BH is short form for that. Then event SD and event TD. So these are other parameters, standard deviations and other calculations which are done in addition to these already available analysis. Now what we need to do is, so if you can see these arrows, you can also sort these values. For example, if I want to see that which of the genes which are expressed in higher level, so simply I can sort it in either in descending order or in ascending order, so that I can simply get the list of those genes which are expressed in higher amount. So I can either select any specific gene, or I can simply means look for these genes and their expressions. Now this is about the what are the log fold expression change values. Now they also tell you the visual means and visual results as well. So this is for the IDH1 association results. So these are all the list of genes and how the expression of IDH1 is associated with other genes. So this is just a comparison. Now this tells you about the log2 fold change, and this is also the these are the p-value significance scores. So in this case, if you see from 0 to 6 and 0 to -6, so some genes which are upregulated and some genes which are downregulated. So this is for that particular purpose. You can see this um, volcano plot. Now, along with the volcano plot, there are also two other heat maps. So one is for the positively correlated significant genes, and the right side is the negatively correlated significant genes. So in this case, if you can see, this one is the negatively correlated genes and these one are the positively correlated genes. So now, which means that if expression of IDH1 will increase, then these genes expression will also increase. That means there is a positive correlation. And in negative correlation means if IDH1 expression increases, then these genes expression will decrease. So in this way, they will provide you the list of genes, and this is the Z-score. So this kind of tells you that how much variability is available, variability is present. So there are two groups, group 0, group 1. So this is a kind of a cluster, you can say, that they have categorized them into two groups. So this is are the binary groups. And then similarly for the negatively correlated significant genes also, they have two groups, and these are the list of genes, and these are the Z-scores. So means if you can see these all multiple cells, we can say, or multiple columns, you can call them columns or cells. So these are the samples. So that's why these from all these samples there are certain values which because in all the samples you won't be able to get a single value from all those samples. So that value can vary. So in that way, in the end, you need to calculate its mean so that you can assign the mean fold change to a gene, because there are multiple samples from where you are collecting data. So the heat map is kind of a representation in which from all the samples what data is collected. So it is just the representation of that data. Okay, so these are the visually means graphical representations which you will find in the link finder. Now the question, so what question they have asked is, so is it clear till now? Okay, so you have asked many questions. Sorry, I didn't see it. So under cohort, we are selecting the group of people data with that specific disease. Yeah, so in each, they have divided cancers into multiple cohorts. So whenever we select any cohort, so we are selecting all the patients from where they have collected data. We do not have control on that, selecting the uh, group of people or samples. So yeah, we are selecting that particular group which are belonging to particular cancer cohort. Yeah, so means even I'm also not that much aware, but these are the gene level analysis platforms. So there are multiple ways in which they have collected the data. So which could either be at the level of RNA or mutation level data. So these are some of the names which are given to the platforms that from where the data is taken. So GA2X is also one such platform from where they are collecting their data. And data type RNA-seq and analysis gene. Yeah, so means what happen is that, for example, if you have the RNA-seq data and you are comparing the expression of a gene, that how many reads you obtain for means how many reads of RNA you are obtaining. So in that way, you can also means means associate that to a gene. You're not specifically worried about which RNA sequence is that or which protein it will synthesize. You're simply means associating the expression of, you are saying that if the expression of RNA, if reads of RNA will increase, then it simply means that your gene is also overexpressed. You are, in this case, you're not worried about which RNA. So that means the data type is RNA-seq, although you have collected the expression of different RNAs, but you are only giving analysis at the level of gene. Means your interpretation is of for the gene. It's not for the RNA level. But means it can vary, because if you are going for the protein level analysis, then what will happen is that you have same RNA-seq data, but now what you have to do is, because you are associating protein information, so for each RNA-seq, you have to means first of all you have to assemble those RNA reads into a single transcript sequence. So all those things will be done, all these means alignment and everything will be done in the end for star alignment and all those analysis you have to do. So in that way, each RNA sequence will be associated to a transcript ID. So after transcript ID is associated, now you will predict, or you will simply check against the already available reference sequence databases for the proteins with the UniProt or other databases. So you will predict its protein sequence, and in that way, you will compare among them that for a single gene, which proteins are expressed, overexpressed, and which proteins are underexpressed. So when you are in means labeling or adding that protein level information, so that is known as protein level analysis. But if you are not worried about protein level information and you are simply looking for RNA expression, you are counting its read, and you are saying that if for a particular gene number of reads for RNA are high, then I will say my gene is overexpressed. So that is what they have mentioned here, that your data is although RNA-seq, but you are performing its analysis at the level of gene. Yeah, because we already know now. So RNA gene conversion means that we are not converting RNA to a gene. We already have the information. We have GTF files. We already have a genome data set. So we have already means we have the tools in which alignment is mostly done. So whenever you obtain the RNA-seq reads, the first step is that you have to assemble those reads to construct a transcript sequence. After assembling, you have to align them, because you should have a reference genome from where you will be able to find that to which sequence they align to. That's why you'll be able to identify to which gene it belongs. So in that case, after getting the RNA-seq raw reads, so what will be these reads? These will be like a nucleotide sequence. In case of thymines, there will be uracils in the RNA sequence. Now what we will do is that we already have the human genome data set. So in that case, we already having the entire sequence of all those genes. Now we will try to align them, the human RNA reads and the human genome reads. Now we'll align where the sequence is matching. So in that way, we'll be assigning the gene information. So as we have seen in that tool also, so integrative genome viewer, what it does is that it tells you the 3-frame translation from both the sides. So in that case, similar thing you can do here also. You can align your RNA-seq reads, you will be able to find to which gene they belong to. So in that way, you'll be able to identify that which gene RNA belongs to. An overlapping sample. So overlapping sample means that um, there are possibilities that one person who is suffering from breast cancer is also suffering from ovarian cancer as well, because these cancers act like a comorbidities. That means one cancer is followed by the another cancer. So you might have heard a term named metastasis. So what happens in metastasis? There are one category is tumors, another is the metastasis. So it means one is benign or other is metastatic. So what happens is one is localized to a single region, and another category is which is kind of traveling, which can move from the primary organ to the other organs. So one such example is of the liver cancers, so which is also known as hepatocellular carcinoma. So what happens in liver cancer is that if you have a breast cancer, so after reaching a certain stage, my cancer can convert to metastasis stage. So in that way, it can move from the that particular region to other region of the body. So in most cases, if you are suffering from breast cancer or nasopharyngeal cancer or means other lymphomas, so those cancers have ability to metastasize, and in that way they will move to other organs as well. So in that way, if you have multiple patients from where you have collected data, and those patients who are means in both the cohorts, if they are present in both the cohorts, so in that way that they will act as a overlapping samples. So they will come under that overlapping region. So that's why this is the overlap. Yeah, so the values which will be less than zero, they will be the down-regulated genes, and the greater than zero will be the up-regulated genes. And zero means that similar expression. So as I've told you that to calculate the fold change, what they does is means normally what is done is that you firstly you keep the expression of a gene in cancer state divided by the expression in normal state. So uh, means if logfold 2 is calculated, because in most of the cases it will be the logfold 2 value, and it is not just simple fold change. So when logfold 2 value is 0, it means that 2 raised to power 0, it will be equals to 1. That means when you will calculate the logfold change, so that means in both the cases the expression is exactly same, so it will turn out to be 1. And when you will transform into log scale, then logfold 2 value will be, I mean, the value which you will be returning will be the value which you, if you will keep on the power of 2, that will give you the main expression value. So in that case, if logfold 2 change is 0, that means expression is same in both the normal state and cancer state. If it is 1, that means expression will be double. That means the score is 2, which means if you divide cancer by normal, value is coming out to be 2. That means this gene is doubly expressed in that state. Yeah. So now we'll be answering this question from the linked finder. So the question was that search the gene name TP53 in the link finder association results and click on the view. Select the appropriate range of the number of wild type and mutant samples. When TP53 is compared with IDH1 gene in case of glioma, so these are the options that means they have given the range. So this thing is it's quite good because they have given the range, because each time if you go through these servers, the data is updated every time, so values can change. So these are the options. Now we will follow these steps and we'll see what will be the result. Now we have to write TP53 here. So as you can see, we have got these results. Now after clicking on view, this was what appeared. So in this, where the data entries were there, so in their search box was also there. So you simply have to write the gene name which you want to find. So in this case, they have asked us to enter TP53. So we have entered TP53. Now it will open up the list of all those genes which are having this particular word in their name. Now in this case, TP53 is also available, so we'll click on view. So if we click on view, then it will open the bigger window because in the beginning we have selected attribute to be IDH1. So now the comparisons of TP53 will be done in means comparison to IDH1. So now the box plot which appears, it will tell you how much expression variability exists among the IDH1 and TP53. So first thing is log fold change value. So it is minus 0.3097. So this is simple log fold change. This is not log 2 fold change. So this is log fold change and it is minus zero point 0.3, which means this gene is down regulated. So this one interpretation we can make. Second thing is its p-value. So we also have to check its p-value, because if p-value is means e minus 0.6 means 10 raised to power minus 6. I hope you're aware of that thing. So if this value is not e minus 0.6, and what if it is simply 7.90, that means it is insignificant. So that's why p-value is important, because it tells you that the results which you have observed, either they are significant or not. So in this case, they are significant because the value is less than 0.05. Now the sample size is 657. Now the 657 sample size is further divided into two categories. So one category is 0 and another is 1. Now in this case, 0 means it is a mutation data. So 0 means wild type and 1 means mutant. So 0, if we talk about vital status, so 0 means alive and 1 means dead. So this kind of tells you that how many are wild type, how many are mutant. So those are means similarly for vital status also 0 and 1 are used in this case. So zeros is n means number of means all the values. So in these from 657 samples, 248 are wild type and 409 are mutated or mutant, and these are the box means these are the distribution of all those values. So means what are the expression values. So this is just for that representation, the box plot. Now the question which they have asked was to distinguish among the wild type and mutant. So keep this thing in mind that wild type are 248 and mutant are 409. Now we have to see that to which value these fit into. So among all these options, wild type fall in this range between 220 to 280, and mutant type fall among 380 to 440 because they were 409. So in all other options, means 409 is not found. It is only found here. So the right answer will be option D. So I hope it is clear. It was quite straightforward. Yeah, so now let's move to next question. Now they have asked what is the log fold change value for PRLHR between wild type and mutant. Now what we have to do, we simply have to repeat the same thing which we have previously done for the TP53. We simply have to write PRLHR. So now in the search box I have entered this. It will simply give me one such target gene result. Now I can also find log fold change value here, but we can also simply click on view, and this will be the visual representation of box plot. And if you are not interested to go through box plot, and then you can simply find it log fold change from here. Means this view is only if we want to access the wild type and mutant information as well. So in this case, log fold change value is 4.624. So we know that if value is positive, that means the gene which are in question, that is overexpressed, that is expressed 4 times. So now in this case, PRLHR is upregulated because log fold change value is positive and regulated in mute. So in this case, if we compare it, so its expression is higher in case of the mutant type. So that's why the answer was it is upregulated in mutant state, and log fold change value is 4.6. Yeah, it's greater than 4.6, sorry. Yeah, now the next question was that click on view overview of dataset and present in the link finder, and how much total data is FTR adjusted and have value less than 0.01. Now this for this we have to go back and we have to click on overview of dataset, because in the previous two questions, we are simply selecting the gene IDs and gene names. Now we have to look for the entire data set. So simply click on the overview of data set. Now this will give this particular summarized view. Now these are the p values and these are the FDR, means pie chart, which distribute that how the different values are distributed. So in this case, uh, FDR less than 0.05 total 15754, and less than 0.01 is total is 14581. So now let's look at the question. So they have asked that uh, have values less than 0.01. So we have to tell the percentage. So these are the percentage representation, and we have to tell for this less than 0.01. So orange, it is 72.5%. So the right answer will be 70 to 75%. Now next question is download the heat map of negatively correlated significant genes in link finder tab to answer the question. Now they have asked that which of the set of genes are negatively correlated with IDH1 gene in glioma. So now we have to find this. So in the previous, when we were looking in through the link finder, we have seen that in the representation, they also give you the negatively correlated gene set as well as positively correlated gene set. So they have asked us to download it and then look for these genes. So keep one thing in mind that from all these genes, you have to find each and every entry, because what can happen is that in some cases you might find three genes and fourth gene might not be there. So in that case, that option will be wrong. So don't be in hurry in that case. So now these were the two type of genes, positively correlated, negatively correlated. So now we have to download this negatively correlated gene set. After downloading, this particular image will be downloaded in a PNG file. Now those from those options, we have to find it that which genes are available here. So I've already means found it. So in this case, the genes which are highlighted in red color, these were the genes which were negatively correlated and other genes are not present. So in all these four options, there are certain genes which are present in this particular gene set. So you have to compare it for all the genes which are present in each option. So in this case, in option D, all these genes are present in the list, in this particular list. So in this case, option D is the right answer. Yeah, so this was all about this particular link finder tool. And related to your assignment, if you find any problem, then you can simply go through this particular manual. In this particular manual, which they have provided, exactly same problem is discussed. So in this, they have also taken exact same example, and ultimately you'll be able to find your answer also. And you can simply just cross check if you have done with this question. So it might help. Okay, so no. So log fold change and log two fold change are two different things. So when we say log two fold change, we mean that whatever value we will get, it will be kept on the power of 2. But when it is simply log fold change, then it will be on the power of 1. So that is the difference between them. So whenever log fold change is written, okay, where are you talking about? Okay, so in this case, you have talked that they have written log 2 FC is 4.624. Okay, let me check where they have written it. And this log 2 for change. Yeah, in this case, they have not written it to be log 2 FC. But actually, if it is not written as log 2 FC, then it is not considered to be right. It is considered to be simply log fold change. So yeah, in this case, log FC means log 2 FC only. But if you find it anywhere else, then don't take it same. So by default if I think in maths or in other things, when you write log FC, then it is log 10 of FC sometimes, and not log 2 actually. Okay, so less than 0.01 FDR is the valid group. Yeah, because your FDR should be as low as, means if it is more, means if lesser the value of FDR, more significant it is. That means. Okay, so might be they might have, means interchangeably use log FC and log 2 FC. Yeah, so in these cases that might be true, but otherwise these are two different things. Okay, so means is it clear? Should we proceed? Okay. Yeah, so I think for these you just means make sure that these are same. If the values are same, then this means they have used it interchangeably. Otherwise, it's not always the same. So now the next question is, what is the primary goal of pathway enrichment analysis? Option A, to identify individual gene mutations. Option B, to predict protein structures. Option C, determine if a set of genes is overrepresented in a known biological pathways. And option D, to sequence DNA directly from the samples. So now you have to tell the goal, and yeah, it's quite straightforward that pathway enrichment analysis is to done to determine that which set of genes are overrepresented in the known biological pathways. Sorry. So next question is, what is the primary purpose of LinkedOmics? So option A is to create gene mutations. Option B, to provide a database linking multiple level of omics data, that is genomics, proteomics, etc. Option C, to sequence the entire human genome. And option D, to identify protein-protein interactions only. Yeah, so this is also straightforward question. Okay, so now option B is right. So option A says to create gene mutations. So in LinkedOmics, we are not creating gene mutations, we are studying. So that is gene mutation dataset is already available and we are just studying. We are trying to associate the mutational information with the expression level. So we're not creating gene mutations. And to sequence the entire human genome, so no. In LinkedOmics, we're not sequencing human genome and to identify protein-protein interactions only. So this is also not true. Whereas there are other in link interpreter, we are associating the information, pathway level and other level information. So from there we can get the protein-protein interaction information, but this is not the only thing. So option B is the right one in this case. Now question number 9. So in pathway enrichment analysis, what statistical test is commonly used to determine overrepresentation of gene sets in a biological pathway? So you have to tell about the statistical test. First option is Fisher's exact test. Option B, Wilcoxon test. Option C, Z-test. And option D, Kruskal-Wallis test. So you have to tell about the commonly used one. Yeah, so Fisher's exact test is the one which is commonly used to determine the overrepresentation of gene sets. Now we'll see that why this test is used. So in the context of pathway enrichment analysis, our goal is to find whether the set of genes, so when we say set of genes, which means that those genes which are differentially expressed or either they are mutated. So we are trying to phenotypically rank them to a particular set of genes. So whether it is overrepresented in a particular biological pathway compared to what would be expected by chance. So in that way, we are comparing them. So Fisher's exact test is ideal for analyzing the small sample sizes. So if your sample size is small, then this test is better. It works with the categorical data to test for the independence between the groups. And if we talk about the pathway enrichment analysis, so we can often categorize genes as either being part of a pathway or not. So it is a kind of categorical output that either it is a part, either it is not. So it is like a 0 1 kind of thing, and it is not giving you any continuous value in point something something. So that's why it is mostly used, and then see whether a high than expected proportion of differentially expressed genes fall into that pathway. Now the question is that why Fisher's exact test is used. So option means one point is that it is a known parametric test. So it doesn't assume normality, which is important when we are dealing with often skewed distributions. So when we say any test to be non-parametric, then it means other tests which are parametric tests, they assume that your distribution will be a normal distribution. That means values are evenly distributed across the mean. But when we talk about differential data, then in that case, that means there is a possibility then that certain genes might be overexpressed or underexpressed. So we can say there might be more number of outliers, or your distribution can be skewed towards any of the corners. In that case, we require some test which should be non-parametric so that it cannot, means it should be able to capture the variability. Then works with the small sample sizes. So unlike other statistical tests which require large sample assumptions, because if sample size is small, then we are not much confident enough that either our results are means right or either they are just randomly generated. So in that case, this Fisher exact test, it also statistically performs well with a smaller sample size. So that's why it is opted. And the third thing is that it generates 2 by 2 contingency table. So this is the contingency table when we are means this is a tabular representation. So in this case, we have two phenotypes. One is differentially expressed, other is not differentially expressed, or we can call them as a category. So it is present in a pathway, it is not present in a pathway, and we are simply checking the correlation among these columns and rows. So it involves binary categorization. So in this case, either in pathway or not in pathway, and either differentially expressed or not differentially expressed. So these are the values A, B, C, and D. So means this is just variables they have written A, B, C, D. Otherwise, in your data, if you have the data set, then you will be putting those values in this table, and then you will be comparing them. That which of the genes which are present in pathway and differentially expressed, and which are the genes in pathway but they are not differentially expressed. So we will not consider those genes which are not differentially expressed. Means for further interpretation and analysis. Now, Fisher's exact test calculates whether proportion of genes that are differentially expressed in the pathway, which is this cell A, is significantly higher than the expected ones. So that is the next step when we check that either those enrichment scores or those things which we have obtained, either they are significant or not. So that's why Fisher's exact test is used. If we talk about other tests, so which was Wilcoxon test. So this was also one of the tests which we can use. So it is also a non-parametric test for comparing two groups of continuous data. So in Fisher exact test, we have the categorical data, or you can say we have the binary data. So categorical data not necessarily means binary, but categorical means that it is not associated with any numbers. It is associated with some categorical values that yes, no, high, moderate, low. So that is a categorical data set where the output will be in that form. But if we have a data in which the output is in the continuous data form in some numbers 0.001 or those means expression values, so they are some continuous data sets. So in that case, we use the Wilcoxon rank sum test or Wilcoxon test. So it is used for the ranked data and not for the categorical enrichment analysis. So that's why this option was not right. If we talk about z-test, so this is also for comparing the proportions or means. So if we have a large sample size, so z-test is a parametric test which assumes that the distribution is normal. So one reason that it assumes normal distribution so that we cannot select it for differential analysis. Second thing is that it requires the large sample size. So due to that reason also we cannot use it. And other thing is that it is used for the comparison of means. That among two groups it will compare their mean to check that either they are variable from one another or not. So how much they vary from one another. So if there is significant difference between their mean values, that means they are different distributions. So that is about the z-test. And then comes Kruskal-Wallis test. So this is a non-parametric test and it is used for compare more than two groups of continuous data. So this is also for continuous data and if we have more than two groups. So in that way, it is not applicable for the categorical data. So that's why Fisher's exact test was the well suited for determining if gene set is significantly overrepresented in a biological pathway. So we have already seen. So the two main properties were that of its non-parametric nature and it handles small data set. So that's why it is opted. Now let's move to next question. So the question is, what does GSEA rank in a data set for identifying enriched gene sets? Okay, so we already have discussed similar question with previously. So option A is genes by their alphabetic order genes by their chromosomal positions, genes by their differential expression levels, and genes by their molecular weight. So in this case, you have to tell that how it ranks the data set. Okay, so actually option B is yeah, so in MSQ it could be option B as well. But commonly when we say, so it is generally for the differential expression levels. But in their paper they have mentioned that for chromosomal positions, biological function, and expression and regulatory values. So these are three properties for which, means keeping those things in mind, we can rank the genes. So if your analysis is on the basis of synteny based analysis, then you simply categorize them on the basis of chromosomal positions. Otherwise, by means commonly people use it for the differential expression levels. So to check the expression enrichment of genes on the basis of the expression. So option C is the most appropriate option. Now next question is, in GSEA, a positive enrichment score suggests that, so this is also somewhat related question only. The gene set is mostly underexpressed in the phenotype of interest. The gene set is randomly distributed across the ranked list of genes. Option C, the gene set is mostly overexpressed in the phenotype of interest. And option D, there is no significant enrichment for any phenotype. Yeah, so it's also straightforward that there are two enrichment scores, one is positive, one is negative, and if it is positive, that means it is related to overexpression. And if it is negative, it is the down regulation or under expression. So next question is, which of the following is not an enrichment analysis tool? So option A is GSEA, option B is David, option C is string, and option D is blast. So now you have to tell which one is not an enrichment. Yeah. So right option is blast, because we already seen that blast is the basic local alignment search tool. So which is generally used for the alignment of sequences. If you have a novel sequence, you can put it in a query box, and then BLAST has already defined data set from where it will try to find out that to which gene or to which protein sequence that query sequence belongs to. So this is for alignment and not for enrichment analysis. Whereas if we talk about GSCS, it's dedicated to enrichment studies. If we talk about David, it is also for the pathway analysis. So in that way, to identify those pathways, it performs the enrichment analysis. Similarly, in string also. So this is also for the network protein-protein interactions and all those analysis. So in this also, it performs the enrichment studies. So that's why all these options were right. Now next question is, in GSEA, what is the normalized enrichment score used for? Option A, to adjust the enrichment score for the size of gene set. Option B, to rank the genes by their chromosomal locations. Option C, to calculate p-values for individual genes. And option D, to normalize the protein expression data. So now you have to tell that, okay, so what is the normalized enrichment score used for? Why we have to normalize the enrichment score? So the right answer is to adjust the enrichment score for the size of gene set. So this we have previously discussed, because if we have different gene sets and the number of genes among those gene sets are variable, then in that case we need to normalize the enrichment score. So we normalize that enrichment score is actually the means when we are calculating the enrichment values, it is not the expression data. So we are not normalizing the expression data, we are normalizing the enrichment score. And enrichment score calculation is based on the comparisons among the gene which you have in your gene set, which are present in sample and which are not present in sample. And then you are comparing it with the probability, that what is the probability it can present in this particular gene set. So in that way you are trying to rank them. So that's why it is different from the expression data, and generally normalization is done to balance the sample size bias among different gene sets. So that's why it is done. Yeah, so when we see the heat map, so in that case, in that heat map, we are shown those log fold change values and the expression level variability. And when we say enriched set, so gene set, enriched gene set, and those log-fold chains, so they are different concepts. So in enrichment score, it is a score based on different means phenotypic associations, or you can say in that way. Actually, I'm not able to get your question. So in statistics, size, the population size. So in journal, if for normalized enrichment score, okay, so okay, you are asking that whether we are normalizing it with the size of population or what. So in this case, yeah, so in this case, it is normalized for number of genes which are present and not for the patients or not for the person or not for the population size. Because in population, when we are means for the gene set, it is actually for the number of genes which are present. So for total number of genes, we are normalizing it. For example, if I have 10 genes and enrichment score is 20, for example, so I will normalize it by 10. So 20 by 10. And for example, if I have 5 genes and I have score of 15, so I will normalize it by dividing 15 by 5 so that I get the normalized score. In that way, means for each gene, what is the average? Means I am calculating kind of mean thing so that I can normalize it. Yeah. So next question is that in proteogenomics, what is the key opportunity presented by the mass spectrometry based proteomics determining genetic mutations? Option B, identifying novel protein coding genes and splice variants. Option C, mapping gene regulatory networks. And option D is improving DNA sequencing depth. So you have to tell that in proteogenomics, what mass spectrometry based proteomics helps you, means what help it provides you. Okay. So option B is right because it identifies the novel protein coding genes and splice variants. Because in proteogenomics, we are linking genomics and proteomic information. So in that way, we are able to capture the splice variant information as well. And because it is mass spectrometry, so this provides you about the protein coding gene information as well. Whereas if we talk about determining genetic mutations, so genetic mutations we can't find out on the basis of proteomic level data from mass spec, and mapping gene regulatory networks. So these all things are not related to mass spectrometry. Now next question is, which of the following is a limitation of proteogenomics in clinical applications? So option A is difficulty in detecting mutations. Option B, lack of standardized databases for proteins. Option C, high variability in sample preparation for mass spectrometry. And option D, inability to sequence DNA. So in many cases, there will be some options which are confusing, which appears to be right. So in this case, this is the MCQ, so you have to select one option. Okay, so yeah, so the right option is high variability in sample preparation for mass spectrometry. So this is one of the limitation, although there are some measures which we can opt for and with which we can reduce this variability and we can make sure that sample bias could be reduced as much possible. As we already know that in mass spec, we have designed some methods which could be multiplexed so that we are saved from this sample preparation bias kind of thing and difficulty in detecting mutations. So although in proteogenomics we are not it is not that much difficult to detect mutations. It is easy to detect mutations, but it is difficult to detect the driver mutations. So that is one difficulty, and then lack of standardized databases for proteins. So this is also not exactly true, but we cannot say that it is completely wrong because we lack databases for proteins. For example, they are not means for novel proteins as we say. So the proteins which are not yet structured or not yet for which we do not have the sequence information yet. So we lack that information. We do not have the 100% protein sequence information. So this option is also means a little bit of a limitation, although we have databases available, but it lacks some little bit of information till now also. And inability to sequence DNA, so this is completely wrong. We already have sequenced the DNA, and even currently we have sequenced the telomere sequences as well, and even the Y chromosome as well. So sorry. So next question is, how does proteogenomics enhance our understanding of cancer biology? So option A is by improving the RNA sequencing techniques. Option B by identifying tumor-specific protein variants and their functional impacts. Option C by enhancing CRISPR gene editing accuracy. And option D by increasing the speed of genetic mutation detection. Okay, so you are asking something related to previous question. Sample preparation is from different tumors. Yeah, means it could be different tumors also, and it could also be from the same tumor as well. For example, you are having sample size is huge. There are multiple patients from where you are collecting samples. So when you are preparing your samples, so if in all those samples, if the conditions are different, so in that case also the sample preparation bias will be incorporated. It is not only for the tumor types, but also for the different samples. Yeah. So now you have to answer this question that how does proteogenomic enhance our understanding of cancer biology? So you have to go through these options and you have to tell that what advantage proteogenomics provide us. Yeah, so it is also quite straightforward that it identifies the tumor specific protein variants and their functional impacts. If we talk about by improving RNA sequencing techniques, so proteogenomics has nothing to do related to RNA sequencing techniques. If we talk about the analysis and then finding out the unique isoforms or novel sequences, then it is true, but it is not improving sequencing techniques. Then by enhancing CRISPR gene editing accuracy, so this is also not true, and speed of genetic mutation detection is also not true. So option B is the most appropriate option. Now next question is, what role does proteogenomics play in drug discovery? So option A is it reduces drug development time. Option B, it helps identify protein targets altered by genomic mutations. Option C, it improves the stability of drug compounds. And option D, it provides new techniques for high throughput screening. So you have to tell about proteogenomics. Yeah, so this option is also quite straightforward. That it helps to identify the protein targets altered by the genomic mutations. So we have already seen that how this information is useful. So even if we talk about gene set enrichment analysis, or even we have seen in LinkedOmics also, so this all analysis are somewhat means have the application in the drug discovery or in related to cancer specific studies. So what they are doing is they are trying to associate the mutations to the cancer or to the states, that which mutation is linked with which particular state. So what actually overall is done is that we look for the genes which are overexpressed, then we find out in which pathway they are expressed, what process they are involved in, then we look for their mutations, that if this particular mutation happens, then how this mutation is linked to its expression. Either its expression is decreased or it is increased. So for example, if I take example of the tumor suppressor genes, so these are the genes, if they are expressed, they will suppress the tumor growth. So any mutation which will happen in the tumor suppressor, it will be loss of function mutation. Because if there is loss of function mutation, which means that my tumor suppressor gene will not be able to function. So mutations can be both gain of function or loss of function mutations. Some mutations can increase the expression, some mutations can decrease the expression. So in that way, ultimately what we do is we try to find out that how the mutation is linked with expression and in which pathway that gene is involved, so that overall we can compare, means we can associate them, we can integrate that information, and we can find out, so we can analyze that how this particular gene is leading to cancer. If we know that, then we will be able to find out which place to target. So if one particular mutation is involved, then we have to simply design a drug which can target that mutated region and which can either replace that particular region or which can find out that what is the role of that particular position. Either it is lying near the phosphosite, so which is specific for the kinases and other bindings. So that means depending on that particular context, we can design the drugs. So in that way, proteogenomics is helping us. So option B is the right one. It improves the stability of drug compound. This is also not true. It reduces drug development time. So to some extent we can say, but we cannot say that it will always reduce drug development time, but it because it helps us to identify the targets. So in that way we can design the drugs with lesser amount of time comparatively, because instead of designing the blind drugs, means blindly designing the drugs, it can reduce to some extent time, but not means exactly true option. And it provides new techniques for high throughput screenings. So this is also not true because it is not providing us any new technique. It is simplifying our analysis, but means this is not true. Okay, so next question is, which of the following is a major challenge in proteogenomics? Okay, so these questions are nearly similar to one another only. So option A is limited availability of genomic data. Option B is difficulty in identifying novel protein isoforms. Option C, lack of statistical methods. And option D, sequencing errors in DNA. So now you have to tell which is the major challenge. Yeah, so major challenge in proteogenomics is the difficulty in identifying novel protein isoforms. So if we talk about the genomic data, then its availability is not limited. It is available, and vast amount of genomic data is already available with us. Then lack of statistical methods. So no, we already have lot of statistical methods. And sequencing errors is also not means that much major challenge if we talk about the difficulty in identifying novel protein isoforms. Now next question is, in proteogenomics, how are novel peptides identified? So option A is by comparing RNA-seq data. Option B by de novo peptide sequencing from mass spectrometry data. Option C by using CRISPR gene editing. And option D by microarray analysis. So now you have to tell in perspective of proteogenomics and novel peptides. So it is a MCQ. Yeah, so the right answer is option B by de novo peptide sequencing for mass spectrometry data, because we have already seen that from mass spectrometry data you will get the information of all the peptides, and then you require some database through which you can align your peptides, so that so there is one term which is known as peptide spectrum matches, with which you have a spectrum and you assign it a sequence of peptide sequence. So in that way we require some database which is already available. So in case we are dealing with some novel peptide sequence, so we require the already available RNA-seq data, so that which we can customize to a protein sequence, and then we can identify the novel peptide. But in this question, the first option was by comparing the RNA-seq data. So this option can be taken in two terms. One thing is that by comparing the novel peptide data coming from mass spec with the RNA-seq data, or does it mean that we have different RNA-seq data which we are comparing? So this option is quite confusing. It depends that how you take this option. So in one perspective, it could be right also, because by comparing to the RNA-seq data, which is customized and converted to protein sequence, so we can identify it. But in this is an incomplete statement. So that's why this option is not right. And similarly, CRISPR gene editing is also not, since it's not the technique for identifying novel peptides. And microarray analysis is also not for novel peptides. It is for the gene level or DNA level analysis, not for the protein level analysis. Okay, so now next question is, why is a permutation test used in GSEA? So option A, to normalize gene expression data. Option B, to calculate the p-value of enrichment scores by randomizing phenotype labels. Option C, to estimate false positives. And option D, to adjust for gene length biases. So I think when we were going through the gene set enrichment analysis paper, then we have seen that we require to perform the permutation tests. Okay, so now let's see. So the right answer is option B actually, because permutation test, permutation combination. So I hope you are aware of this permutation term. So what we does is that to identify that whether our enrichment score is significant enough or not. So this is for the second step of GSEA. So in the first step, we have seen that we take the data set of both TCGA as well as TCGA as CPTAC data, both we have taken. We have identified enrichment score. After enrichment score is identified, second step is to find that whether that enrichment score is significant or not. So in this second step, we will perform the permutation test. What we will do here is to find out the significance of ES, we will repeat that we randomize the samples. So in this case, we will randomize the phenotypic labels and we'll just again perform its enrichment of those genes. And we'll see that either that enrichment is appearing randomly or either that is significant enough. So this permutation comes under step 2, which comes under the significance of enrichment scores when we are identifying the significance. And when we say significance, that means there is some p-value which is given. So to calculate the p-value, and if we talk about to estimate false positives, so this false positive comes under step 3, which comes under multiple testing correction. So this will not be, means in this case, we do not use the permutation tests. And then to adjust for gene length biases, so we are not going for this gene length bias adjustment. So for permutation test means that you are randomizing your data set. So we are not, means permutation is a thing where we randomize it. And when we are linking the test to it, that means you are randomizing it and you are calculating the p-value. So permutation test is different thing. P-value calculation is different thing. So permutation means randomization. So randomizing phenotypic labels means permutation test. But in this, after randomization, what you are doing is you are calculating p-value. Yeah, so p-value calculation is based on the other statistical tests. By performing the permutations, they are calculating the p-values. Okay, so okay, so when we are trying to identify the p-value, what we does in p-value is like finding the significance that whether the sample and the background, so in the sample and the background, how enriched the sample set is. Means the genes which are present in the sample set, either they are enriched or not. So if I will change my sample, I will randomize it again and again. So in that case, p-value will also change. Yeah, so in that way they are related. That randomizing will influence the p-value as well. Okay, so this was the last question of this session. Okay, so now I hope next week or maybe next to next week you will be having your exam. So good luck for that. And if you have any questions related to this course, then you can ask. Yeah, I've already seen your query related to Morpheus. So even I also tried with Morpheus. So what is happening that each time it is giving some, it is, yeah, so in k-mean clustering, it is showing random result each time. Each time the samples or the columns, they are also even changing. So the two questions, first question was related to that means clusters and the number of patients in each cluster that you won't be able to answer because each time it is changing. And the second question, which was also difficult to find out, was the from there you have to select one of the patient which is not present in the group. So each time it is changing. So I've also seen the same thing. And even I tried to go through their manual also because in manual generally they explain each and everything. So in manual they have not even explained k-mean clustering. They are only talking about the hierarchical clustering. So generally if we perform any such clustering methods or any such approach, Then there is one field which you might have found, which says that how many number of repeats or I don't exactly remember that thing. So it was like how many number of times, number of batches, or maybe something like that. So in that, by default it was 1,000, but in the assignment they've asked you to do it 100. So that batch size means the number of times those batches are done. So that is also one of the thing which can lead to change in the values, change in the number of clusters and how the values are clustered. Yeah, so this is the real problem. You can ask them and they should give you some points for this because each time results are different, so you cannot answer it. Even I tried it, means I had tried to find out. So one time it's giving the right answer, other time it's giving the other answer. So I was not able to figure out one single answer for it. I even tried increasing that value also from 100 to 1,000 to 10,000 also, but each time it is giving some different clusters. So what I will do is that in the discussion forums, I will also write the same thing. Actually, I was also waiting for the official TAs to address that question. But they have not addressed it so far. So what about other questions in the assignment? Because in other questions also I've seen that they have mentioned the institute name and they have not exactly mentioned that what should be the data type or what should be the platform exactly. So were you able to follow that thing of LinkedOmics? Yeah, so that's good if you were able to find it out. Okay, so for Morpheus, they should. Yeah, I will mention it. Actually, I do not have direct means contact with them. Otherwise, I would have asked them. Okay, and one more thing which I want to tell you that last time you have asked about the SNPs and SNVs. So that single nucleotide polymorphism and single nucleotide variations. So those two terms are different because when we talk about SNPs, they are single nucleotide polymorphism. So how we assign SNP to one single nucleotide is that we have a populations. So after comparing the people in the population, we assign SNP positions. So in all those people, that particular nucleotide is varying at that particular position. So it is assigned as SNP. If this nucleotide is SNP, that means in multiple populations, it is polymorphic, it is changing. But when we say SNV, these are the single nucleotide variations, and it does not mean that it should be present in all the population, it should vary in all the populations. This could either be a mutation, which is not common in the population. So they are referred to as single nucleotide variations. So all the SNPs, they are SNV. That means all the single nucleotide polymorphism are single nucleotide variations, but all the single nucleotide variations, they are not SNPs. So this is the one thing which I have found out about the SNPs and SNVs, and I think which I have also incorporated in the previous slide as well, this thing. So if you have gone through that presentation, then you were able to find it out. And thank you so much. So both of you, because you two were the people who were there throughout the sessions. Otherwise, means I would be not motivated enough for these classes. Yeah. Okay, so if there are no further questions, then I'll end this session. And good luck, both of you, and all other students who might watch these recordings later. So, okay, then. Thank you so much. Goodbye, good luck. Good luck for your future because we'll not be meeting for the next session. So, bye bye. Thank you.